Okay, so let us look at this. Mitochondria, as we know that double membrane bound organelles only present in eukaryotic cells. Prokaryotic cells, they don't have mitochondria, but they do have the enzyme carry out the respiration. So be careful about this, okay? So they don't have a special organelles here called mitochondria for prokaryotic cells, okay? So they have two membranes. So each of them, they are phospholipid bilayer, and outer membrane, they are smooth. Inner membrane, they are convoluted, or we call it the fold. They are infolding, forming what we call the cristae. Okay, so the space that the space between these two layers of the membrane is called intermembrane space, and then the one that's surrounded by the inner membrane is termed as mitochondrial matrix. Now be careful, I prefer you guys to use the word mitochondrial matrix because the words here, okay, sometimes question will ask you, okay, the reaction take place where? If you say matrix, then it's not correct because a lot of place you have matrix, we have the what we call extracellular matrix. Can I see that? So which part of matrix? Which is mitochondrial matrix. So the matrix contain many different enzymes, okay, as well as circular mitochondrial DNA and 70S ribosome. So this enzyme involved in the aerobic respiration. For example, enzyme that involved that we have learned ATP synthase that synthesize ATP, and also we do have other protein known as electron carrier that functions during this oxidative phosphorylations. Okay, so this is general overview. Let me show you guys more detailed parts, and then I will conclude for these reactions, okay, and also the mitochondria. Okay, so for just go for the brief one first. So for respiration, aerobic respiration, involve 4 stages. Okay, so the first stage is termed as glycolysis, followed by the second stage called link reactions, and then the third stage is termed as Krebs cycle, and number 4, oxidative phosphorylation. So means that whatever we have learned previously in terms of the chemical from reactions that we see that C6H12O6 plus 6O2, the oxidation process here, we get, okay, 6CO2 plus 6H2O plus energy release, okay, plus ATP. So even though we can see that it's a single step process, but this is a net process, means that it's involved, okay, in this process, and this aerobic respiration actually involve these 4 stages. Are you clear? Glycolysis, link reactions, Krebs cycle, and oxidative phosphorylation. Now, glycolysis actually independent of oxygen. Means that whether O2 is there or not, it doesn't matter because glycolysis still can take place even though without O2. Are you clear? Now, don't say that without O2 because glycolysis also can take place with the present O2. So I will use the word independence of O2. So it means that whether O2 is there or not, it won't affect the glycolysis. And this glycolysis take place inside the cytoplasm. So nothing to do with mitochondria. So in this case, nothing to do with mitochondria, yeah? So whether mitochondria is there or not, glycolysis will continue to take place, okay? But these 3 reactions, link reactions, Krebs cycle, and oxidative phosphorylations take place inside mitochondria. And in fact, we need to know, okay? In fact, we need to know the precise locations of each reactions, okay? So link reaction take place inside the mitochondrial matrix. Okay, Krebs cycle also take place inside the mitochondrial matrix. Okay, and oxidative phosphorylation take place at the mitochondrial inner membrane. Are you clear? See here. So means that three of them, okay, these processes must take place inside the mitochondria, and they have term and conditions. What is term and condition? Term and condition means that the conditions must be aerobic condition. So under aerobic conditions. So means that if let's say under anaerobic, basically the reactions won't take place. If under anaerobic conditions, then glycolysis take place because glycolysis is independent of O2. Are you clear? Glycolysis independent of O2, so means that they will continue to take place. Are you clear? Even though under anaerobic condition. So when under anaerobic conditions, then you can see that mitochondria stopped carry out link reaction, Krebs cycle, and oxidative phosphorylation. We're going to learn why. Okay, why only aerobic respiration? Why only under aerobic condition mitochondria can carry out link reactions, Krebs cycle, and oxidative phosphorylations. Okay, so highlight this. It must be under aerobic condition. So therefore, when you learn about this mitochondria in your first chapter, we always say that it is a site for aerobic respiration, because anaerobic respiration won't take place inside mitochondria, right? Anaerobic respiration never take place inside the mitochondria. So what is the function of aerobic respirations? Okay, to produce ATP. Are you clear? To produce ATP. So this ATP produced through two processes, which is known as substrate link phosphorylations or substrate level phosphorylations, and as well as oxidative phosphorylations. Are you clear? Substrate link phosphorylations and oxidative phosphorylation. Are you clear? Clear? So it means that in order to carry out these reactions, means that mitochondria must have a certain ability to do this, okay, or adaptation. So let us look at this. So first of all, mitochondria, outer membranes, they have a smooth outer membrane. Okay, outer membrane. So write down the characteristics, smooth. So what is the role of outer membrane? What is the role? So basically, outer membranes separate. Okay, separate what? Separates the environment. Okay, separates the internal environment. Okay, mitochondrial environments and cytoplasm. Okay, what they do? They separate the internal environments of mitochondria and also cytoplasm. Okay, so what are so special about these outer membranes? So these outer membranes, they have, okay, they have the karyoprotein. Karyoprotein of what? Now, few karyoproteins that we can actually, write down here. So first karyoprotein, pyruvate karyoprotein. Okay, karyoprotein for pyruvate, reduce NAD, as well as ATP, because ATP is formed inside the mitochondria. So we must have a karyoprotein to bring the ATP out of the mitochondria. Okay, so now at this moment, don't worry about those pyruvate reduced NAD. When we finish our aerobic respiration, we come back, then you understand already. Automatically one, no worry. Are you clear? So no need to memorize at this moment, because since we are talking about this mitochondria, so might as well tell you guys this. Okay, clear? Okay. Now second layer. Okay, second layer, then you can see that we have the inner membrane. So inner membrane actually is highly folded to form cristae. Okay, so label, this is the inner membrane. So inner membranes, they are highly folded, forming cristae. Okay. So if you point to cristae, how you label a cristae? Then you label at the folded regions there lah, okay, for cristae. So what is the functions of this inner mitochondrial membrane? So inner mitochondrial membrane involved in oxidative phosphorylation. Okay, they're involved in oxidative phosphorylations. So what kind of protein they have? They have a lot of protein and enzyme that embedded on these inner membranes. Okay, they are protein and enzyme embedded. So what are the protein? The protein involved here is the electron carrier, for example. Okay, electron carriers. Let me write down better. That form electron transport chain. Or called ETC. Okay, what are the enzyme involved? They have the enzyme that involved here known as ATP synthase. And both this protein and enzyme, they are involved in the oxidative phosphorylations. Okay, now still remember when we talk about oxidative phosphorylation, we need to accumulate, right? We need to accumulate proton at the intermembrane space, right? Can you see that? So you want to accumulate these protons at the intermembrane space, you need this electron carrier. Okay, so we're going to learn detail in these oxidative phosphorylations why electron carrier is so important for this oxidative phosphorylation. ATP synthase, we know about this. Okay, ATP synthase involved in the formations of ATP. Okay, through oxidative phosphorylation. Okay. So now the third part here, okay, is termed as intermembrane space. Okay, formed because of these two, right, between these two layers of the membranes, and these two layers of membrane form this intermembrane space. So the role of the intermembrane space here also involved in the oxidative phosphorylation, but to very, very minor extent. What they do, they accumulate proton. Okay, so in terms of structure, they are very narrow. Okay, they are very narrow. So why narrow is so important? Because when they are narrow, in this case, so means that the area, the volume smaller, concentration higher. So therefore, because of narrow, we can create this steep proton concentration gradient. Or we can say that a steep electrochemical gradient. Okay, this intermembrane space. So what the role here? The role here is to accumulate protons. So make the proton concentrations very, very high. Okay, we can create, but in order to make the concentration high, because we know that concentration is the mass over volume, so means that the volume must be low, then concentration will be high, right? So therefore, you can see the intermembrane space actually is a very, very narrow. The space is very, very narrow. So therefore, it can create this steep proton gradient across the intermembrane space and the matrix. Are you clear? Okay. So the last one we're going to look at here is the matrix. Now, matrix or mitochondrial matrix have a lot of function. Okay, so first of all, in this mitochondrial matrix, what we can find, basically we can see that they have a circular DNA. Okay, they have circular DNA. So what's the function of this circular mitochondrial DNA? Sometimes we add the word circular mitochondrial DNA. So what is the role of circular mitochondrial DNA? So circular mitochondrial DNA actually contains the gene. So this gene actually code for enzyme or protein involved in aerobic respiration. Are you clear? So they contain the gene. So this gene will undergo what we call as a transcription. We transcribe the gene so that we get the mRNA. So this mRNA later, you can see that inside the matrix of the mitochondria also have the 70S ribosome. So this 70S ribosome going to translate, what they do? They're going to translate mRNA. So the translation of mRNA going to form the polypeptide. And this polypeptide is going to fall into enzyme or proteins. So what are the proteins? For example, protein, electron transport change. What are the enzyme? ATP synthase, for example. Okay, so those protein enzyme involved in the aerobic respiration again. Okay, and the third, in the matrix, you can see that we have a lot of enzymes and also proteins that involved in the aerobic respirations. Okay, so this is a summary for the mitochondria. Okay, if you learn about mitochondria, you cannot run away from this. Okay, so what are the reactions? Sorry, guys, let me add in something. What are the reactions involved in the matrix of the mitochondria? So mitochondrial matrix involve these two main processes. One is called as the link reactions. Sorry, link reaction, not link. And Krebs cycle. Okay, involved in link reaction and Krebs cycle. And then this oxidative phosphorylation take place at the mitochondrial inner membranes. Okay, you can take a photo, you need. Okay, so we look at these ultra structures of a mitochondria now. If you look at this, so we can see a lot of cristae or the infolding. Can you see that? So this infolding increases the surface area. Okay, so what's so important about to increase the surface area? Because we do know that we at this inner membrane we're embedded with, or we have this inner membrane can accommodate the enzyme ATP synthase, also accommodates this electron transport change, right? Okay, the electron carrier. So therefore, you can see that, okay, increases the surface area. When you increase the surface area, definitely you can accommodate more protein, more enzyme, so that the rate of the ATP synthesis will be high. Okay, and you can see that the inner membrane and outer membrane form this a very narrow space here known as the intermembrane space. So this intermembrane space is to accumulate the protons, okay, so that we can generate a high concentration of proton, or what we call this, we have these steep proton gradients. And then you can see that the dot, can you see that the dot? So those dot actually, they are matrix of the mitochondria or mitochondrial matrix. The functions here is very important because they do have a lot of enzyme, they do have the circular DNA and also the ribosome, 70S ribosome, not ATS, 70S ribosome. Okay, so with this, I have done for the first part of today's lessons. Do you guys have any questions so far?